biosafety labs, which are at the heart of the controversy surrounding the origin of the virus. We could not have found two more capable people in the to do just that. Both are my friends and colleagues. They're not only first-class active scientists, but are also exceptional human beings that are fighters for peace and justice in our world. So our moderator today is Jonathan King, a famous molecular biologist, longtime professor at MIT. Jonathan's brief bio is more than a page and a half. So you can imagine it's an exceptional career with unbelievable achievements, not only in science, but in other areas of our existence. He is a professor of molecular biology at MIT, where he taught biochemistry and directed biomedical research on protein folding for many years and won many prizes and, and honors. Professor King is the author and co-author of some 250 refereed publications in his area of expertise with grants from NIH and other US national institutes. He currently serves on the board of Citizens. This is the other side of these two scientists, that they are not only just scientists, but they are also great human beings who are trying to make our world a better place to live for the future. And so I won't go through all of these accomplishments of Jonathan, but I would say that he had made a tremendous contribution to the adoption of the legislation in the United States in 1984 about the biological banning research on these types of viruses and so forth. Although later we did not have that ban, there is a loophole, and he will talk about that. But there was a tremendous contribution that Jonathan made. And he is also a leader of the national campaign, as I said, of biomedical scientists on the BWC in 1984 with the adoption of the legislation. Outside of his tremendous academic and other achievements, he is now devoting most of his time in the non-academic time, that is, as the co-director of Massachusetts Peace Action in its fight against nuclear weapons around the globe, and also against militarism and unbelievable military spending that we are undertaking in this country. So without much further ado, I will turn over to Jonathan, but just before that, welcome my friend Monica Zoppe from Italy. I know we had a little bit of trouble with the Zoom, but I think we're all set to go. So thank you everybody and welcome.
Thank you, Subrata, for the very kind introduction and for organizing this series, and to Trish Weinman and the Technology and Culture Forum for its support. Now, Cambridge is a particularly appropriate setting for investigating biohazards, since it was here in 1976 that the Cambridge City Council held the first hearings on whether the newly emerging gene splicing technology pose novel biohazards either to the laboratory workforce or the people washing dishes in the laboratory kitchens or the surrounding population. If so, then new public health regulations would need to be developed. Those of us who were microbial geneticists involved in developing the technology understood that it would be historically transformative. We also knew it would rapidly give rise to a new manufacturing and production technology. Indeed, some of the Harvard scientists arguing against regulations had already applied for patents and were setting up the first biotechnology companies when the hearings were held, which we learned from the Wall Street Journal. A number of us concerned knew of the long history in which corporations whose products had toxic effects actively worked to suppress that information becoming public. This was already well documented lead poisoning from lead paints, smoking and lung cancer driven by the tobacco industry, asbestos and mesothelioma, bladder cancer from aromatic amines produced by the chemical industry. But for genetically engineered organisms, for example, bacteria or viruses, there was the additional risk that the products of the new industry, microorganisms, were self-reproducing. We have very few examples of biological pathogens, such as invasives, such as Dutch elm disease or chestnut blight or Japanese knotweed or new influenza strains being removed from the ecosystem once established. So there was an extra dimension of concern. Now, as everybody here knows, the outcome of the intense debate was the establishment of safety guidelines for laboratories conducting gene splicing research. These are now used throughout the world. Business analysts have concluded that one of the reasons the biotech industry boomed here in Kendall Square, Cambridge, was that the establishment of the regulations, despite the industry's resistance, established a level playing field through which the new companies could reliably navigate. However, though the proponents of commercialization were willing to accept regulation, they argued, falsely I would say, that since the research could be carried on safely, there were not intrinsic hazards. It is as if physicists argued that since experiments with radioactive isotopes can be carried out safely, therefore radioactive isotopes carry no hazards. Not true. An unfortunate result of the debate was the complete neglect and in fact total suppression of research on the possible biohazards of genetically engineered microorganisms. Those of us who thought there were hazards, we proposed experiments to the NIH which could reject or accept hypothesis, and they declined. They never established any kind of research or epidemiological effort in that direction. So that was the situation for many years. It changed after the 9/11 attacks on the two towers. The Department of Homeland Security launched a multi-billion-dollar program based on the assumption that terrorists could use genetic engineering to develop new and novel pathogens. Homeland Security advocates argued that the US needed to explore such organisms in order to develop adequate defenses. Now, unfortunately, the Homeland Security never refers to these threats as biological weapons, but uses obscurantist language such as dual-use threats. I think most of my colleagues are not aware of the level of kind of hazardous research supported by Homeland Security. Such programs have been established around the nation, but because they're all covered by national security concerns, it's very difficult to find out the details of the research. As many Boston residents learned during their opposition for many years to the building of such a facility in the heart of downtown Boston, which now does exist. Now, many questions have been raised to the origin of the SARS-CoV-2 virus. We're lucky to have Dr. Monica Zoppe addressing these questions. She is unusually well prepared for this discussion. She's been on the faculties, the International Center for Genetic Engineering and Biotechnology, the Clinical Physiology Lab of the National Research Council, Italian National Research Council in Pisa, and the Institute of Biophysics in Milan. Her research included cellular studies on transport of viral vectors for gene therapy. So she's been an active member of the Scientists Against War group in Italy, constituted in the 90s, in which she has been especially attentive to the theme of biological weapons, the responsibility of scientists, and issues of dual-use research. She's also an active member of the Pugwash community, which still exists, and has participated in national and international meetings dedicated to the construction of peace for a just and equitable world. And we're so happy to have her, someone who's willing to speak candidly about these pressing questions, given the worldwide devastation of the coronavirus. Monica, please.
Thank you so much, both Subrata and Jonathan, for your very, very kind introduction. and for organizing this series of meetings, which I think is very important at this time in the world. And yes, indeed, Jonathan touched some of the important points that I will address during the presentation, which I hope it will meet your expectation. As a forward, I want to say that I also followed the previous presentations in the series, but this one is going to be quite different because before we met with the situation in which two opposing sides, India and Pakistan, the USA and the Russian Federation, were facing each other. What we now face in this situation is one of the great challenges that the entire humanity is facing. Not that the nuclear war would not be a problem for the entire humanity, but this, the challenges we face now with the COVID-19 and with other global issues must be faced by the full humanity as a single entity. So in this respect, what I think is that the COVID-19 case has shown an unusual capability of putting together all scientists in the world and doing and all of us working together, facing this problem, sharing all the information we get as we get it. And despite this being a very minor episode, it bodes well for the future, I would say. Not that this is an insurance, but hopefully it will be something that will become more usual in the future, and it will help in the solution of all of our problems. Now let's get into the talk. I will share the screen. Does it work?
Yeah.
Okay,
Yes.
great. So what are we going to talk about? It doesn't go on. Oops. Oh, here. Oh. I apologize. I'm using a different. Okay, let's go back. Okay, so I will just make a short introduction because I'm not sure that everyone in the audience is familiar with some of the terms that we are going to use, like biosafety, biosecurity, select agents, and so on. And then I will talk about the high safety laboratories and how mistakes and accidents happen. We'll get some details, not too much, but some details will be explored. And what's as a conclusion to this part, we'll talk about the transparency and preparedness on issues like new diseases, emerging diseases. And finally, I will also face the issues raised about the origin of the COVID-19 pandemics that we are facing right now. Okay. Okay. So to start with some definitions, when we talk about biosafety, we have something that is referred to the safety of the operators and of the community, of the environment, and is related to the dangerous danger linked to the organism that that people are working with. So this can be bacteria, virus, or other pathological forms. But the important thing is that no cures are known for this organism. Biosecurity is a term that applies to the control of what is being done in the laboratories, which may not be a high-level biosafety laboratories, on the personnel that is working on them, and it implies a set of regulation that must be followed. This is something that is especially an item, a definition that is that originates with the United States of America government, is not a worldwide definitions. And indeed, the rules and the regulations that apply to the biosecurity, that is to the people that are allowed or supposed to be working with bioweapons and select agents, are a set of regulations that are decided and apply only in the USA. So when we talk about bioweapons or select agents, again, we consider a biomaterial that can be used in an inoffensive attack. And its important features are the lethality, the amount of the material that the people are handling or preparing, and the weaponization. That means the means of delivering to the supposed enemy. In between the two, there is the problem of the dual use that Jonathan also mentioned, and that is indeed a problem that arises regularly when work is done with viruses that can infect humans or animals that are or plants that are important to humans. So the select agents, as I was saying, are a category that is defined by the US government based on national security considerations. So this is a military categorization. There's more than 60 of them. They are controlled by the Department of Agriculture or the Department of Human Health, or both. However, some of them are frequently occurring in the USA or elsewhere in the world. For example, cholera is an endemic disease in much of the old world. It is now also become an endemic disease in Haiti. That was a very unfortunate situation. Another obvious agent which is present in nature is the Ebola virus. However, we should not be confused, because the select agents are not necessarily worked in a high-level security lab. We will see the biosafety level 4 is considered the highest level security lab in the highest level. So let's see the so-called BSL4, Biosafety Level 4 laboratory. The image here shows a schematic. This is from Wikipedia. It's kind of a summary. Basically, all of the BSL4 laboratories are custom built, so there's not one the same as another. They can be rather small and carried on a truck, like, for example, some of the research labs that are being used during the epidemics or the Ebola epidemics in Africa. This is the smallest BSL-4 laboratories in which people don't even enter. They're just the boxes in which people can work. Sorry. Oh, okay. But some of them can be very, very large, and their cost is really, really huge. They proliferated after the BioShield project that was started by USA President Bush after the 9/11 and the same time anthrax attacks that happened in the USA. And so the project BioShield was like, I don't know, number of billions, but the proliferation of the BSL-4 labs is going to cost more and more because the very simple maintenance of these laboratories can be in the tens of millions per year, excluding personnel, material, just the keeping these laboratories empty but active. So working in laboratory of high safety is not very comfortable. People wear suits which are under positive pressure because so that any damage to the suit would not allow entry of any material from the air. They breathe air that is coming through different route, so breathing in and out. Anything that goes in or out in a BSL-4 laboratory is under strict control. It's filtered once, at least twice, autoclaved, sanificated, well, bleached. And people have to go through showers and cover, so the laboratory are under negative pressure, so that anything a breach would not, and so on. So it's some of them are so large that they can even host hospital beds. But not just that, they can have animal facilities or agricultural facilities like greenhouses, all enclosed in a biosafety level lab. The biggest of the BSL-4 laboratories in the world, not surprisingly, belongs to the US Army, military, and is, well, 835,000 square feet, which in square meter would be 77,000. This is more than 500 times a regular 4-bedroom apartment. So basically this building, well, this is a series of buildings which include possibly 5, 10, maybe more high security laboratories. This makes it actually difficult to know how many of these laboratories are in the world. And this is one of the first secrets that is surrounding these laboratories. So I have tried to make a catalog, well, to know how many of them I could find. And not surprisingly, most of them are in Europe and in North America, but there are some also in basically all countries, all continents, sorry, for a total that is estimated to be around 70 high-level biosafety level 4 laboratories in the world. They belong to the universities. About 30% are in universities. Just as many are in military, so they are almost exclusively used for and by military operations. There are also some private high-level safety labs, which are some for the pharma industry, but also some private, like in Texas, there is a BSL-4 lab, a very large BSL-4 lab that was built as a foundation by an oil magnate. He decided to do so, and it's a private enterprise that is probably receiving also public money, and with link with the university and the military, because of course the final entity that is involved in BSL-4 labs is the government. Governments of or at different levels, that state or local or federal or the other institutional government health research. They are all now marked in a circle, because it is very often the case that the things are a little bit mixed. So some military research is done in university labs, some private research is done in university labs or in governmental labs, and it's very fluid situation, because it's whoever wants to do high-risk research tries to find a place where they can do it, and according to how much they want to have it publicized, they may choose one or another. But why do we want to know how many they are? Because the number of laboratories is it tells us how many people are allowed and have both training and access to dangerous material. So this is many thousands of people, scientists, technicians, other personnel that have access to BSL-4, but also BSL-3 or BSL-3 enhanced, so-called laboratories, and have access to dangerous material. Not only there are so many, large number, but the turnover of the people that work in these places is quite fast. This is not surprising, because working in such laboratories is very stressful, and people can't do this job for a very long time. So there's a very, very large number of people that have access to dangerous material. I repeat, because I think this is a very important point. So, but then why do we have these laboratories? As we said, in the highest level labs, the organisms that are under study are those that that are the most dangerous of all. That means that they have a high rate of human transmission, and they are highly mortal, and for which no therapy or vaccine are available. You can see probably some of the most known Ebola, Marburg virus. Variola is the agent that causes smallpox. There is a vaccine, but it's not available because it is supposed to be eradicated. So there should be no variola, well, variola major at least, circulating anywhere in the world. This was a virus that was exclusively human. So once eradicated, that should not exist anymore. Mistakes happen all the time. Humans make mistakes based on several causes, because they are not skilled enough, because they are just arrived in the lab, they want to do something that they're not properly trained, they may not know exactly what they're doing. However, the classification of mistakes is usually the list that we see here shows that they are basically unavoidable, because needles and sharps are always used in the labs. You may substitute them as much as you can, but when you have to inject an animal, for example, there's no substitution for a syringe with a needle. Spills or drops from culture plates are, well, they are not so frequent, but they happen. And especially when you work in the conditions that we have seen before, which are not comfortable at all, they may happen with more easily. Then samples can get mixed up. One may be in a hurry, just to place a plate in the wrong level in an incubator, and the next worker comes in and takes that plate instead of the other one, and the samples get mixed up, and a mistake happens. Finally, also, this is also quite frequent, when people work with animals don't like to be injected with diseases, of course, and they tend to react by biting or scratching the researchers, which, even if they are covered with the several layers of gloves and sleeves and so on, sometimes it happens. I have mentioned here, and I will not talk about that anymore, about intentional acts. This is something that we know can happen. The anthrax episode in 2001 in the USA, when mails, when letters were mailed containing actually highly weaponized anthrax spores that led to the death of a few people. This was obviously something intentional, and it was originated in a high-level security laboratory in the United States of America, and I think it was demonstrated that the anthrax spore were derived from Fort Detrick. The person that committed that, well, the supposed person that sent the mail was a worker in the lab. However, he, after several years, he committed suicide, but really never confessed. And a definitive proof that he was the perpetrator has not been presented. So it's possible to doubt that the case was solved. However, let's not talk about intentional act anymore, because we're talking about mistakes. So mistakes happen. The conditions are difficult, the place is stressful, people become fatigued, they can have personal problems. They can have just, they might be in a hurry because they have something to do afterwards, and they might be slightly more sloppy than usual, especially when they are confident enough because they have done the same thing several times. Maybe it happens. We all do these things. When we are in a hurry, we make mistakes. That's a normal thing. Mistakes are just the first steps, because when people make a mistake, then they try to fix it. The first thing they do is they try to fix it for several reasons. Because if they get caught, they might be reprimanded. Because if they reveal the mistake, they might be stopped from continuing the work. And that may be some work that have taken months to plan and to start. Sometimes people just don't really adhere to safety regulations, but it's very easy that one simple mistake starts a chain of events. When many people are working in the same place, one drops a plate, another one walks on it and slips on it, and then things start getting confused, and what was a simple mistake become an accident. Now, what happens when an accident occurs? The most likely outcome is that it is hidden, that is institutional cover-up. Just a selection of some episodes that posed risk for that arise from mistakes. I see it's a little bit late, so I don't want to go through all of them. Maybe I just skip this all and leave it to you. If some of you is interested, then we can go through them afterwards. Well, same for this, because the avian flu, a few years ago, about 10 years ago, this was something that was in everybody's. Everybody was concerned about this. It was the avian flu, famous avian flu. We don't have the time to go through all the details of this case. However, I just want to stress that finally we have made a worse strain of this flu. The avian flu is very dangerous. It's highly lethal. But it was only killing birds and occasionally jumping to humans, but directly from birds. So there was a very rare transmission among humans. After a lot of discussion, a report was actually 2 reports were released in 2012, in which researchers in Europe and actually Kawaoka, I'm not sure if he was working in the United States or in Japan. However, they selected a highly, a form of the same flu, highly transmissible in mammals. So you can imagine an accident in which a mouse, a small mouse really this big, escapes, and it is bearing avian flu adapted to mammalian. I don't want to think about it. Another case study that some of you may know and that you might be interested because I know you are from Boston, um, is the Boston University building of the high-level biosafety lab that Jonathan was mentioning before. Here again, I will not go through all of it, but in November 2004, well, some accidents happened. In November, the university got approval for building the high-level safety lab. And at that time, they informed the public authority about the accident, the incident that had happened months before. However, the public was only informed months after that. As it was recalled before, this started a strong public opposition. This is also interesting because investigations revealed lax behavior. So this is blaming the victims. It's a very common way of getting through of not solving problems, but placing them on the shoulders of the victims, especially women are familiar with this situation. However, the construction was initiated in 2008, and the first work at the Biosafety Level 4 laboratory was started 10 years afterwards. So let's just skip. This is the building, you may recognize it. Accidents. Human makes mistakes, but non-human accidents also happen. This is a graph showing the number of accidents of intervention that the select agent programs had to do relative to natural accident. The list is not updated. In 2019, the number seems to be slightly lower. However, as you see, most of them are linked to the weather and to the environmental problems, and these are set to grow. Yeah, there's nothing to do. Well, there would be something to do, but we are not doing that. So basically, the climate will change more and more. Extreme episodes will become more frequent, and so there will be more disruption to normal work in any situation. Earthquakes are not so frequent. They are not linked to climate change, but in this case, we have to remember that the Fukushima accident happened following an earthquake. So the power plant in Japan was built to resist any earthquake. And in fact, it did resist the earthquake. What was not forecasted was the following tsunami that basically made the damage that was not made by the earthquake was made by the tsunami. So even if we can think about accidents, we can try to prevent damage brought about by natural accident. It's always difficult to predict everything. Just like the earthquake generated a tsunami, fires may generate explosions. In the labs, there are gases, flammable and explosive gases that are sometimes used in laboratories. An explosion may happen in following a fire. So this there are chains of events which are totally unpredictable. So what are we going to do with the high containment laboratories? The risks are there, risk for the personnel, risk for the community, risk for the environment. Are they really necessary? This is a question that is legitimate to ask. And my opinion is that in some cases, it can be good to have them. The reason which is the most common is the study of emerging or re-emerging diseases. Now, if they are re-emerging, then like the Ebola, for example, we keep going on that because it's the most spectacular, I would say, even if this is not a word that we want to use for such a deadly disease. But it is, there are good reasons to study that virus, and that virus must be studied in a very, very controlled situation. Emerging new diseases, we have right now an emerging new disease, and no research done in any of the high safety labs is helping in the pandemic. When the pandemic started, we had no vaccine, we had no medicine, no drugs, we had no way to face the disease. So the claiming this reason to justify the building of high-level containment labs is not necessarily justified. Bioterror containment is totally wrong because these BSL-4 labs, rather than contain, they may help any bioterror action that people may do. We have seen before the number of people, the availability of the material, the mistakes that happen, the intentional things. So there's no way that a research that produces weaponized agent can be used, can be of help as a containment of bioterror. And finally, this preparedness, we don't know what for, and this, once again, this COVID situation that we are now shows that there was no preparedness for nothing whatsoever. So bottom line, risks and accidents do occur. Oops, I had a mistake. The necessity for high-level containment labs is limited if it is there at all. So how do we do? My personal opinion is that we should have just a few laboratories, less than 10, all over the world, distributed geographically, coordinated by some supranational authority like the World Health Organization, because they should be only dedicated to real diseases that are important for human activity. Diseases that might also be diseases of animals or agricultural diseases. But the important thing is that they should be controlled. The number of people that work on this in these places should be reduced as much as possible. They should be from everywhere in the world. They should be international, because that would also help to build trust, and each one of them should be trusted. Now, this trust is something that's not so easy to define. However, any research program should be under strict scrutiny by the community. That means that what happens in these laboratories should be fully transparent. People should know what happens in the laboratories. This for two main reasons. One is that because people know, they finally may trust the fact that these studies are made for the human good. And second, because if accidents happen, and they do happen, then to exactly know what was inside, which diseases, which organisms were in the lab and may have escaped, this will be very important to prepare for to face possible leakages. So this is my personal stand on the biosafety, high biosafety level laboratories. Now let's go on and talk about the SARS-CoV-2, COVID-19. So I will not resume the situation. We all know it's very bad. It's more than 1 million people died already. Every day there are 300,000 new infections worldwide. And this is a number that is increasing. It is a bad disease. It is not as bad as some other disease that humanity has faced before. The Spanish flu killed between 20 and 50 million people in 1919. And that was when the human population on the planet was about 2 billions. That means that 10 to 20% of the people died. Before that, historical records about the Black Death or the plague, they tell us about diseases that killed up to 30% of the population in the areas where they hit. So it is true that the COVID-19 situation is bad, but in my opinion, most of the damage is an economic damage. I'm not thinking of reducing the importance of people dying. I know it's a very sad thing, but the mobilization that is being put about in this time is mostly about the economic damage. And I will also add that most of the economic damage is in the rich countries. However, the economic damage, although it is mostly in the rich countries, is impacting more on the weaker side of the population in the countries. So, well, it is important to study and to stop this pandemic, but let's just keep it in a perspective that is slightly larger than the today, because that's what's the situation. This graph shows the emerging infectious diseases. This review is from 2008, but the situation is not changed much in qualitative terms, at least. As you can see on this side, most of the new diseases are due to bacteria, but we don't care. We don't worry too much about bacteria because we have antibiotics, and most of the antibiotics are active. And new antibiotics are sometimes found. Of course, there is the problem of the multi-drug resistance, but it's not well, it is a problem, but most of the new diseases are bacteria that are sensible to antibiotics. On this side, what we see is that most of the new diseases found are of zoonotic origin. That means that they come from animals. It's a perfectly natural thing. It's always happened. It happened from the 40s, that since we have started studying the origins of the diseases. So it's it was expected. Well, that the pandemic was expected has been said so many times. I think the most famous claim was by Steve Jobs, but before Jared Diamond was preaching about a new pandemic that would hit humanity. And before that, the WHO warned about it since at least the 90s, if not before. So it's something that, despite the fact that we were expecting it, we were fully unprepared. Some example of the new diseases of viral origin. Just quick, just go through this very quickly. I only highlighted a few, the four that are coronaviruses. Other are viruses of other retrovirus, filovirus, other families. Well, the first one everybody knows was the SARS in the 2003-4. Then there was this coronavirus that was found in Hong Kong, HKU1, in 2005. One person came down with pulmonitis, lung disease, and the virus was recognized as a coronavirus. After that, the virus was found in many, many, many instances. It is one of the common cold virus. So it was recognized after one person had a serious disease, but most of the time, this virus does not cause very serious disease, just a mild cold. Then there is the Middle East respiratory syndrome, the MERS, that affects camels, dromedary and camels, and that can sometimes skip to humans, but again, human-to-human transmission is very infrequent. Maybe, well, just once or twice it's been recorded, and now we have this new one, the one that we all know very well. Now, why we have such an abundance of coronavirus, and why most of the new viruses originate from bats? Well, there may be some bias because we know them better than others, but bats are mammals and are flying, so they can spread around very easily. But also, there are more human reasons. That is that much of the territories that bats inhabit in nature are being disturbed by deforestation for agriculture, by urban expansion, by people seeking wild animals in the forest, so the more we get in direct touch with these wild animals, the more the chances are that one of the diseases makes the jump into humans. So, where's the origin of this SARS-CoV-2? The most salient genetic features of this virus are all found in the spike protein. The spike, I'm sure you know, is the protein that sticks on the outer layer of the virus, and it is responsible for binding to the cells and to cellular receptors. So the three features are the receptor binding domain, furin cleavage site, and some glycosylation sites that we will just look in detail one by one. So the binding to the angiotensin converting enzyme 2, this is a protein that is found on the surface of all human cells, and actually of all or most cells of any mammal. Now, it is a very ancient enzyme. It is very conserved, and many mammal, well, the proteins from different animals are all very similar because they have evolved for such a long time that they have now reached an equilibrium, optimal equilibrium, I would say. They are very similar. They can be exchanged between species. The receptor binding domain is the part on the spike. If we consider the spike this club that stands on the surface, the receptor binding domain is just a minor part on the surface of the spike, and this RBD seems to be extremely well adapted and can bind very, very efficiently to the human receptor. The SARS of 2003 also bound to the same receptor, but the sequence was different. So here, I'm not sure you can see it, but maybe we can't, we don't want to get too much into details. However, there's a number of amino acids, not very many, that are responsible for the binding. And those that are responsible for binding to the human receptor in the SARS are different from those that are responsible for binding in the SARS-CoV-2. So this means that the two viruses are not derived one from the other. The most immediate known homologue of the human virus is this bat virus, rat TG13, that is very similar all over, except in the receptor binding domain. The receptor binding domain, the highest similarity found in nature is with this virus that is adapted to the pangolin. The pangolin is quite a small mammal, very peculiar. Nobody knew about it until last year, but now has become famous. So how is it possible that a new virus acquires the mutations that make it so highly specific and so strongly capable of attaching to the receptor? Well, because it's happened before once, at least with the pangolin, that means that in nature it can happen. If it's happened once, it's possible that it happens twice. One possibility is that there might have been some recombination if an animal that was hosting the two different viruses made a new one in which a part came from one and from the other. This recombination among viruses happens, and this may explain the presence of this domain in the SARS-CoV-2. The second feature is the so-called polybasic cleavage site. This is the acquisition of 4 amino acids, is actually 12 base pairs that are not present in any of the most similar coronavirus related to the SARS-CoV-2. And it actually has a role because it increases cell toxicity. So that probably, this is not yet completely explained, but it may be responsible for more lethal or more important disease, because the presence of this site primes the spike to enter more easily. But this is so strong that it happens before new virus is made. And basically, what happens is that a cell that expresses this protein on its surface before the virus is ready to be shed may fuse with neighbor cells. So this is called cell fusion and is highly damaging for tissues. So it is it is a feature of the virus that is important. Now this this feature, as I said, it was not present in the closest relatives of the SARS-CoV-2, but it is present in some other coronaviruses, including the one that we saw before, the one of the common cold, the HKU1. So again, this is something that happens. The selection of cleavage sites that have a similar function in different viruses can be obtained in the laboratory by serial passage of viruses in the cells. These conditions are the best possible conditions for the virus. In the laboratory, the virus is pampered. Anything is made so that the virus can multiply, can proliferate as best as it can. So this facilitates selection of features that facilitate the spread in the laboratory condition or in situation that may mimic the laboratory condition. That is, in places in which the host is very abundant, very close, and available, immediately available. So again, we can ask, does this feature come from spontaneous selection, or does it come from a serial passage in the lab? The two possibilities are both possible. I mean, there's no way to know for now. But before we go to the how to find out, let's just see the final feature that characterize this virus is the presence of sugar chains or oligosaccharides. This is a word, is a difficult word to say, sugar chains, that are made available by the presence of this proline here. This one, once again, is something that is not present in the other viruses. It's it's been acquired possibly together with the cleavage site. And proline is a particular amino acid that induces a kink, a bend in the amino acid chain, and makes it possible for the sites close by to get sugar side. Now these sugars, what's their function? They can be selected. Well, the reason why they are selected is that they possibly hide the epitopes from the immune system. What does this mean? That this feature, besides the fact that it's very hard to engineer without knowing what will happen, but to select for this feature, you need to have a live animal, mammals with an active immune system. This cannot happen on a petri dish. Of course, serial passage can be done in animals. Serial passage is basically what we are doing, passaging the virus one to the other right now, because we are very, very frequent, very susceptible, the best condition for a virus to proliferate. So here, well, there is some studies can be done about the speed of
Time is getting,
mutation. Oops, Jonathan?
the time is getting tight.
Okay, sorry. So I will just be fast and well, I was basically what this study is saying is that the speed of mutation that it's been observed in the coronavirus as isolated from humans all over the world, it's more than 40,000 genomes, is mutating at the speed, at the normal speed that that other coronaviruses mutate. So this would somehow argue, even if it's not a conclusive argument, but it would argue against a selection, a laboratory-built virus. So this is my almost final slide. Luckily, I was almost finished. Where did this virus come from? The only way to know is to find the intermediates. So these are either in the wild or in the lab. The only way to know is to do research, to look thoroughly in the places and in the environments where this virus may have developed. The possibilities can be tested, can be investigated. However, this investigation will need the contribution by scientists with very different expertise, with very different background, and from different places. I mean, the Western way of doing research is something that we trust because we always do it. But the Chinese and Chinese way of doing research is similar. It's not that they think differently. However, most of the expertise in the field of coronaviruses is in the hands of Chinese scientists because they have been studying that for a long time. We know they have been studying that also in the high-level biosafety labs in Wuhan. but that's and that's a good reason to get them in the team that study the origin. Of course, full transparency on all sides is mandatory. No one is going to take for good any findings that is not supported by strong evidence. So maybe a role for Pugwash in this enterprise. Of course, the WHO is the most important institution that can lead such an effort. I think I'm done.
Well, thank you.
Okay, now let's skip this. I just leave this on, so maybe if you have any questions, if you want references or any other details available, that's my address.
So,
Thank you so much. I'm
thank
sorry I took more time than I expected, but I hope things were clear.
Invaluable. So there's a Q&A, those of you who are on the call, there's a Q&A function in this particular meeting. If you put your comments or questions in the Q&A function, I'll try to read some of them off. It's only but I don't see any questions. Well, while I'm, let me just second a key point that Monica made. You know, if you don't know what they're working on in the biological weapons and the world of homeland security, you have scientists trying to think up the weirdest, most unexpected hybrids that, you know, some terrorist might come up with. And then they construct that virus or that bacteria in the lab to find out how to defend against it. So these things are going to have very unexpected properties. You know, you can imagine a tularemia that's making a cholera toxin. Somebody in downtown Boston pick up that infection. Local physician's not going to be able to spot it. They have to know what is being used in those laboratories. And one of the great dangers of this essentially biological weapons research is that it's kept secret, so that when an accident does occur, people don't know it, they can't document it. And, you know, the Chinese were great in the sense that they released the sequence immediately when they had it, right? They didn't have to do that. That allowed US biotech and pharmaceutical companies and scientists to get to work immediately. But they have not communicated what are they doing in their high security lab in Wuhan, right? What are they studying there? And, you know, we need to know that. So it's, you know, really going to be a step forward in overcoming, you know, military national security concerns.
Yeah, no, I totally agree. I think full transparency is the only way that that we can keep doing this kind of research, precisely for the reasons that you said, because we must know what happens in the labs in the case an accident happens, and also because of the people will not trust each other if they don't know what they are doing, if the secrets are kept. Right. I agree that it's very bad that we don't know what happened in the Wuhan laboratory, just like it's very bad that we don't know what happens in the Fort Detrick of the Boston or the Texas laboratories, on any laboratories, Lyon, I mean, anywhere in the world. These laboratories do secret research, and it's very bad. I totally consider a disgrace that these research are kept secret.
Okay, one of the somebody Bernie.
This does not mean. Oh.
Bernie Sanders has asked, can you go back to that slide showing the actual documented containment mistakes that you moved past very quickly? Yeah, right.
This one.
Yeah, yes. And maybe, so these are all documented cases. Maybe you could just pick out one or two and highlight them, because it's very important the actual.
Yeah, this is just a partial list of documented cases. There's, for sure, there's many more, and most of them have been hidden. And as I said in another slide, the most likely outcome after an accident is institutional cover-up. This is something that we can take for granted. It always happens all the time, and people don't want that. They say they don't want to scare. They don't want to stop. They prefer that the people would not know how to react. So basically, they just keep it secret. Okay, I'll just go through quickly through these cases. The smallpox cases in 1978, they ended up with two people died. A few people were infected, two died, and the head of the laboratory afterwards committed suicide. That's quite unfortunate. I think he would have better spent the rest of his life to fight for the complete elimination of all smallpox samples that are still present in military laboratories in the USA and in the Russian Federation. However, and we know that now in the year 2000, people are working with live smallpox in BSL-4 laboratories. This is unacceptable, in my opinion. It's totally unacceptable. The Butler saga. This was a very sad story. This is a research. It's a medical research, a medical scientist working on plague. Plague is still endemic in some parts of the world, especially in the old world. And he was doing a genuine research, but at some point, minor accident happened in his lab, and he was basically caught in a security nightmare. He was finally found guilty on not keeping the right tracks or shipping or transporting samples from Africa to the United States, basically in his pockets. Because that's, I mean, when you go in Africa and you find a person that is affected by plague, you can't put it put this person in a BSL-4 lab and transfer this person in the United States. But he was very careful. That he's never been found. His research was valuable research, and that was a terrible story. The Australian mousepox. So Australia has a problem with imported mammals. Australia was the land of marsupials and there were no mammals. There's now but the rabbits and mice are a major environmental problem. So some scientists had this idea of making a pox virus that would not kill the animals, but would make them sterile. So basically, that's why I wrote an environmental research turned bioweapon, because what they found was an extremely virulent strain. But you never know exactly what you will get when you start a research, an experiment. So sometimes things turn out the way you expect, and sometimes they are different. You have to know what you're doing. That's the major point. But basically, they built this kind of bioweapon that would have. Then finally, I think they destroyed it. They never used it.
Well, Sabrata, Trish, we're past time. I don't know what your policy is on how long you want to continue past time.
Yeah, we had, of course, a little late start. So I had asked Trish, she might have had to go to a meeting, but Christina is there. Christina, could we have 5 more minutes or?
Absolutely. Please continue.
Okay. Monica,
Then let's, because there are many questions now. Thank you.
Maybe, okay, if someone is interested in these mistakes, these are all available in the literature. The anthrax, the Sverdlovsk accident, there was a major release in Russia during the Cold War in the USSR. The leaks, well, the 2001 letters full of anthrax spores. And then there was this episode in which live samples were shipped to several laboratories that were expecting an inactivated samples. So these people were handling live anthrax instead of inactivated. They were just doing like calibrating machines that you need in order to recognize possible cases. So that's just an example of mistakes. This didn't
Monica, could you jump to the SARS? It seems to me particularly relevant. I'm not familiar with this case. Twice escaped from Chinese lab in 2004. That's seems to be particularly.
Okay, so this this was I might have made a mistake. This was 2014. I apologize. I will have to check. However, of course, once the SARS small epidemic that hit basically East Asia and Canada. That was the only two places that were affected in the case of the first SARS, were China, Hong Kong, and Canada. And then it was contained. The people started studying this virus, of course, because it's happened once, it may happen again, so it's important to study. The researchers that were doing this study in China became infected. And I think it was one researcher that became infected and went home and infected someone else in the family. But this was immediately contained, luckily. The disease was immediately recognized. The researcher knew he was working with SARS and he could take the appropriate measures immediately. So there was no bad consequences, but the accident happened.
So let me say there are a number of questions now in the chat, but they're not simple questions. And our speaker is somebody who, if you write to her, she will respond. So maybe put back up on the screen your contact information, because I'm sure that many of our people who participated in this are going to have further questions for you, given your willingness to be transparent and to share.
Yes.
So there's the email for all to see, and it works, right? And I would encourage people to continue the discussion. It's a very difficult, arena, but it's very, very important, and we need to, keep pursuing it. So with that, I'd like to again thank the Technology and Culture Forum and Subrata for initiating the series. Everybody on this list will get announcements of the next one, but Subrata, maybe just mention the date of the next.
Yes, the next seminar is very relevant, not necessarily for the biological discussion on biology we had today, but about military research in the United States. And so the speaker will be Heidi Peltier, she'll be speaking about the cost of endless wars since 2000. And I hope many of you will come. And I know this is very close to also Jonathan's heart and Monica's. And I want to really take this opportunity to thank Monica for a tremendously lucid presentation that average people can access without much knowledge of biology. And for having Jonathan here, it's a real honor to have somebody like Jonathan to be participating in our discussion. And so thank you very much. And Trish and Christina, who all helped us do this.
Brother, let me put in one more plug.
Sure.
There are very few places in the United States, including universities, where you can have a candid discussion of issues in war and peace.
Yes.
Military industrial complexes everywhere. They're financing, research fellows who are, tell you that, oh, of course we have to have nuclear weapons. But because of people like Subrata, because of the technology and culture forum, which is quite unique, it's not under the MIT administration. It's under the Episcopal Diocese of Massachusetts, so it's independent. And because of the history at MIT of leaders of the nuclear disarmament movement, Philip Morris and Henry Kendall, et cetera.
And Jonathan King.
Right. This is a forum where you can have honest discussions. And let me tell you, there aren't too many places in the country where that takes place. So
thank you.
Okay, just a final word from me. I just want to thank you, both Subrata and Jonathan. It's been a great honor for me to be invited to speak in this place, and I also thank all the people that have followed this seminar. I'm ready to answer, if I can, any of your questions, and I'll be there for the next seminar. Thanks.
Thank you. Thank you. So I guess we have to wrap up now. So Monica,
yeah.
Have a good evening, and
thank you.
And Jonathan, thank you. Okay, Christina, thank you very much. Thank you.